return to this towards the end, but let me briefly describe what will be covered in the genomics program, and then I will go back to describing what the project-oriented component really is. So for the genomics program, which is an intermediate program, we will cover the basics of genomics, the applications of genomic sequencing data analysis to study a variety of different examples like germline mutations, genetic diseases, cancer, and similar things to that. So briefly to describe what I will do today is I will cover some of those basic things, provide you with some examples, and then at the end, I will go back to this idea of project-oriented training that you can learn in these programs that are going to be coming up. So as you all know, DNA, especially the human DNA, is very complex, and it's organized in such a way that we can understand some of it, but really we need to have all the information about the genome to be able to ask questions about the DNA. Not only that, also the DNA itself is stored in a very compact way. And so to understand what information it contains, we need to understand the organization of the DNA, which is organized by chromosomes. And so the chromosomes are essentially these strings of DNA that are attached to histones. And when we think of DNA, right, the first thing that we need to think about is what are the actual elements that matter, right? Where should we look at? And so where we need to look at is typically referred to as the whole exome, right? So what are those specific regions of DNA that produce protein genes? So what we'll do in this program is we will talk about whole exome sequencing that helps us understand how the genotype, or what's written in the DNA, defines the phenotype of an individual cell, the phenotype of a tissue, of an organ, or the whole organism. Now, it turns out that only 1% of the human genome, about 1 to 2% of the human genome, are protein coding genes. And that number has significantly went down from the original expectations that researchers had when they were starting to sequence the human genome. So today we have about 22,500 genes, but that number varies across different research fields. And so really, we're going to look specifically at that known part of the genome, which we call the exome, right? The part of the genome that is being expressed. One of the basic applications of genomics is to identify bugs in the code. How can we link specific variants or differences from the reference that allow us to make assumptions that the sequence change results in a particular change in phenotype? For example, here you can see how a mutation could change the cell phenotype, which in this case is the red blood cell, to be the sickle cell. Right, so that sickle cell disease is defined by one such change. What we will also look is how we can start characterizing these mutations and start understanding their functionality, how individual nucleotides form nucleotide codons, and how changes in specific positions in these tricodons result in a change at the amino acid level. We will talk about different types of point mutations, like substitutions, insertions, and deletions. And we will discuss what are genetic variants versus mutations versus neutral mutations, pathogenic mutations, and essentially mutations that don't matter. And the way that we will talk about this is we will try to understand the changes at the sequence level, at the DNA nucleotide level. We will then try to understand what are some of the thresholds that are used to understand whether the mutation is a coding or a non-coding mutation, where that mutation is found. Is it found inside the gene, at the promoter of the gene, at the splice junction of the gene, or whether it is silent? And we will also look at the impact of the non-coding mutations and whether these non-coding mutations matter at all. We will also take a look at some examples of where such germline mutations are known to play a difference in phenotype, in how they affect the phenotype. So when we talk about these types of mutations, we will also apply this to somatic mutations as well as germline mutations. And we'll talk about how to find them and what type of sequencing is required to be able to find them in a with a level of confidence that you can actually annotate them and interpret them. So genomic variation that we will discuss will include looking at individual single point mutations, as well as structural rearrangements and copy number variation as well. And as I already mentioned, we will take a look at how we can observe the impact of these mutations in the sense of affecting a phenotype. And that could be either a disease or that could be just a difference in condition. We will also take a look at the relationship between the specific known genes that typically have these mutations at different levels. At the level of where they are found in the chromosome, what does the gene look like in terms of what's being expressed and what's the variation of expression, at the 3D structure of the protein that that gene codes for, and also at how people validate that these mutations actually cause the phenotype or a change in function. Now, to understand all of this at the practical bioinformatics level, we will also discuss how this data is generated. So we'll take a look at different types of sequencers, and different methods of sequencing, including targeted sequencing, amplicon sequencing, and whole DNA sequencing that is applied for genomics. And to really understand the process, we'll take a look at how the sequencing machine works and what type of data does it produce. And when we come to that part of the data, we will actually look at what do these files look like. And so we'll try to understand what is the information that is encoded in this DNA sequencing data, and then how can we analyze this data, which we will start from by learning the logic of analysis. So really, how can we perform alignment? How can we find variants? How can we interpret the variants structurally, and how we can measure relationships between sequences and changes in the sequences. So here are some of the applications that we will look at. We will look at the need to clean or pre-process our data. We'll talk about the need for calling a variant, determining whether this is a mutation, and annotation. How do we actually know what this mutation could be related to? But we will also look at additional applications of different methods where we can either visualize the data that we have or identify a particular type of mutations like germline or somatic mutations. We'll learn to work with genome-wide association studies where we need to understand the relationship in a population to a phenotype and what is needed to make that kind of an association. And we will also take a look at downstream interpretation of the found mutations. So how can we actually know what a particular individual mutation or multiple mutations, what do they mean, and how can we identify other factors associated with that? And so here as an example, we will use a study that shows how we have individual or monogenic SNPs, and how we can look at polygenic SNPs, where monogenic are strong associations that are typically rare, but they are commonly associated with the phenotype, or polygenic scores that are more complex. And how do we actually analyze the data to try and determine what is associated with what? And this is typically referred to germline mutations. So some of the things that you will learn in this part of this course is you will look at how to load the data, how to process the data, how to find meaningful associations, and also how to provide a report on what you have found using both the T-Bioinfo platform and coding in R. Now, beyond those, we will also look at some of the applications of genomic sequencing for biomedical research. And there we will look at examples for drug metabolism, somatic mutations, inherited mutations, rare genetic disease, as well as drug resistance in bacteria, which is going to be a little bit different, but with similar principles. As a part of that, you will learn what goes on behind those typical reports that are provided to cancer patients, for example, and how do these studies that intend to understand the mechanistic or statistically significant mutations, how do they use this research to actually produce such results? Also, we will look at how cancer occurs, and we will use some examples, the commonly known and something that everybody has probably heard about, like P53, as well as less known as and maybe less understood individual mutations or multiple mutations that have high association with a specific phenotype. So we'll talk about point mutations, insertions, deletions, and rearrangements like copy number variations. So I also mentioned that other applications of sequencing might include finding specific mutations responsible for drug resistance, like antibiotic resistance and tuberculosis, or association between variants identified in short sequences. For example, we'll take some examples of viral sequencing data to try and find specific mutations that have been associated with the phenotype. As a part of this training, you will get access to our online coding, which is like an element that you can find inside the coursework, where you can practice how to write code with a lot of guidance, right? So one of the important parts of this program is to provide you with the logic of analysis and then introduce you to practical skills that you can also take with you and continue to learn on your own. So when we will do that, this is just an example of an exercise where we will take several DNA sequences from patients, compare them to the wild type, and try to determine what should be the specific diagnosis based on this information that we should give to a patient. So again, some practical examples of how you can use this type of data and this type of analysis to identify some important information for biomedical research. So in summary, we will provide you in this genomic program, OmicsLogic Genomics program, a variety of examples and opportunities to try to analyze whole exome sequencing data from humans and non-humans alike. We will introduce you to how this type of analysis could be applied in basic and translational research. You will understand the terminology and the commonly used approaches for analysis, and will be able to perform those independently as well, using the platform for logical and user-friendly analysis, as well as learning how to take the process data and extract from there meaningful insights using some coding skills, not a lot, but at least some, in R, and we will also show you how to deal with a variety of different data types that you will be expected to work with as you are introduced to genomic data analysis. So to briefly summarize, the two programs that we discussed today are Introduction to Bioinformatics, getting started with big data bioinformatics, and Genomics. Now, I think tomorrow we're planning to do another orientation session that is going to be dedicated to metagenomics and data science, which are more advanced programs for those that already have some basics in those fields, like you might have some background in microbiology, or maybe you're interested in data science, which will typically require you to have some background with coding. So you must have at least a little bit of experience coding in R or Python, because very quickly programs will become challenging for those that do not have that background. So again, how do you register for these programs if you are interested? This is the page for Introduction to Bioinformatics. So again, I put in the link in the chat for Introduction to Bioinformatics. And this is a page for the Genomics program. So the Genomics program, the Genomics program right here. These are the topics and the syllabus. So you will find here all of the specific details. All of these programs are going to last for 3 months, so for the whole semester. The Introduction to Bioinformatics is much shorter in terms of what we typically expect people to spend on that program, but you can take up to 3 months to complete it. Genomics and metagenomics and data science might require a little bit more time. The process will be that you will have all of the materials, For example, this is a course on this portal that you registered for already on the Omics Learn portal. So here you will find some asynchronous materials, and every week you will have an email with a video lecture that connects these materials into a program. In addition to that, you will have access to our mentors. So if you have any questions along the way, you're struggling with some piece of code, you're trying to build a pipeline and it's not working, or you have a conceptual question, you would like to understand some topic that you don't think was explained well enough, we will have 2 Q&A sessions per month that you can join to ask all of those questions. Now, Farhana is going to be the course coordinator. She's going to be available for collecting those questions and making sure that we have those questions ready for whenever we have the Q&A session. And she will be able to point you in the right direction for registering for those Q&A sessions. What does a program actually look like? Let me just I'll go here. So a program will have, for example, let's go to this genomics program. So this program, as you can see, will have, once you're registered, you will see here the full curriculum, and you will be able to see here the video recordings that are sent to you as well, and complete those. And once you are signed up, you will find on your profile that right here, you will find whatever program that you're a part of. You will find that mentioned right there. For example, here, this is a program that already finished. And once you have finished, you will find your certificate of completion right here. And also you will be sent a PDF version of that program at the end as well. So now for those of you that are from LBRN, LSU, or NAIPI, maybe for Hannah, you can take a little bit more time to just go over the process again